Hello, everyone, and welcome to our second day of our symposium. Hope you enjoy all of the participants and all of the presentations. Dear participants, please welcome our doctor assistants, Dr. Bilgen, Kaymakamzade, who will now give the presentation titled by The Importance of Mathematical Models on Medical Researches. Please welcome Assistant Professor Kaymakamzade.
And hello everyone, good morning. I think you are can see my can you see my screen? I think everything is okay. So first of all, I would like to thank the organization committee to inviting me, such a good organization. Today I will try to explain the importance of the mathematical model and on the medical research. Maybe you can think that this topic is far from your studying, but be sure that I'm hoping that, of course, after this presentation, you can find some area to studying this kind of topics. So first of all, let me give some information about what is mathematical modeling and why we might want model. Sometimes some model can be complex and we need to simplify them. Mathematical model plays an important role to understand the dynamics of the disease. And it gives the best strategy to controlling the disease for a long time. And it helps us to find the best treatment outcomes of the disease and predict the outcomes of the epidemic treatments. If we check these figures, from the right-hand side, you can see the realism of the seams of the real tree. But sometimes I think that you will agree with me. To study with this kind of figure is hard. So it is easy to study with the left side part. To do this, we are using mathematical model. We simplify our complex things and we are doing analyzing. We understand the structure of the disease. Then we can come back to the real part and we can apply our results to the real life. I want to talk about the feature of the mathematical epidemiology. It starts in our life in 17 and 16. And the first model constructed by Bernoulli with studying the smallpox. In 1906, Hammer formulated the discrete time model for measles. And in 1911, with using the ordinary differential equation, malaria, which is the human to mosquito and mosquito to human contact, Rose trying to do some model for this kind of disease, and last in 1927, Kermick and McKendrick create a compartmental model, and nowadays we are using this kind of compartmental model in our life for describe the disease and explain the dynamics of the disease. And this kind of modeling are input and output type of model. Here, first of all, I would like to explain the first model which making the Kermick and McKendrick. They create a model with dividing the population in a three compartment, S, I, and R. S is a suspectable class, I here is an infected class, and R is a removed class for this kind of model. And any person in suspectable class can remove and goes to the infected class when suspectable person with contact with the infected person, then it can be take this disease in body, and individual can remove suspectable class and can go to the infected class as the rate of the beta. And after infected class, we take some treatments and we can recover from the disease. It means that we can remove infected class to the recovery class with the rate of the gamma. Or unfortunately, maybe we don't have the treatment or it's not suitable for us, then we can't live, so we can unfortunately die because of the disease. So it means that we can remove this class with dying, and again we can remove that class, so we can live infected class and we can come to the removed class. Sorry. And with using this kind of structure, we obtain the ordinary differential equation like this. When we construct this differential equation, we use minus to explain the removeness of the compartment, and we use plus for the coming of the compartment. So we say that we remove from the suspectable class with the coefficient of minus beta Si. So we put here minus beta Si. And when we remove this class, we will come to the infected class, which is beta Si. And after coming the infected class, we can remove this class with a minus gamma I, and we can come to the recovery class. And to explain we are coming to the recovery removed class, we will write here gamma I. And with using this kind of model, we are doing some analysis and we understand the structure of the disease, and we try to find treatment, maybe vaccine, or we can predict what this kind of disease will be in the future. Of course, this kind of chemic and mechanic models are studying for when we have the recovery part, but some diseases have not. For example, influenza, smallpox type viruses have a recovery part. So after recover infected part, we have immunity. Then it's slow to take this virus again. But sometimes we don't have this kind of recovery part. So we have to increase ourselves. This type of model need SI type of model or SI type of model. For example, HIV or AIDS type viruses have no recovery after taking the disease. So we have SI type model, so we don't have a recovery. After when suspectable person contact with the infected person with HIV or AIDS, they can be removed suspectable class and they can go to the infected class with HIV. After HIV part, a person can be take some treatment and they can live with HIV and they can stay in that class. Or if they don't take a treatment, or if it is too high in his body, they can remove HIV class and they can go to the AIDS class. After coming to AIDS class, the only removing from the AIDS can be died, unfortunately. So the person can be died because of the AIDS and remove that class, or for all of each class have the natural death rate. So the other part for the removal of compartment can be the natural death rate. And tuberculosis type epidemics have no any immunity. So after taking these illnesses and after recovered from the infected class, we can go to the back to suspectable class because we can again contact with the infected person and we can take this disease again in our body. So this kind of model need SI type model. And some model have more than one strain, so we are saying that more generally multi-strain modeling. And for example, West Nile viruses have two suspectable population, mosquitoes and human. So we need two strain here. Or sometimes viruses can have mutation. So if we have mutation, it means that we have two type viruses. So as you can see in that model, we have two type infected area. If we have two type of infected area, it means that we have two part of recovery if diseases have the recovery part, of course. And after this kind of model, we can talk about another type. It's same, the structure of the modeling is same, but it seems a bit different from the disease illnesses, cancer or cell type of the modeling. Again, here we can divide our compartmental model and we can understand the structure of the cell and we can create our system. This is this model for making for the apoptosis color 3 to 9, and we are hoping that this kind of model publish will next month. This is our new model, we are studying on it. Now, after giving the some basic explanation what is the modeling, I need to explain what is the basic reproduction number. So maybe you are hearing the basic reproduction rate number or ratio, both of them can be used all of the world. As we know, nowadays we are fighting the COVID-19 and to fighting of these viruses, we are always saying basic reproduction ratio. So careful, basic reproduction number is greater than 1. So we have to use the rules more, or we are saying that, okay, our not values start to fall down, so we are going well. But what is that number? That number is shown with R naught, and it is the number of secondary infections that one infected person would infect in suspectable population. So I mean that if this number is 3, it says us one infected person can be infect 3 other persons. And here, when we are here, if one person again can be infect other more 3 persons. So if we continue like that, as we can see, we have a huge number of infected persons, it will be. So we must careful about the disease because we have a pandemic here. So what it would be if this number was going to be 2? It means that one infected person can be infect 2 more persons. So one infected, this time will be infect 2 more, and other 2 will be infect the other 2 as well. So it will be going like this. Still, we have a pandemic here as well. It's not huge, the previous one, but still we have a pandemic here. So if we say that R naught value is 0.5, for example, this time it means that one person will be infect half person. So it's still the infected person will be goes to fall down. So this is gonna be dies out at the end. We are expecting that. So we are saying that if R0 is less than 1, the disease is going to be dies out. So there will be no pandemic in that area. But if R0 greater than 1 or equal to 1, then there will be a pandemic, and we have to be careful about these illnesses. Here you can see some diseases which they take their place in the literature. For example, MERS are not values of the MERS. It comes 0.8. So it means that one person can be infected 0.8 person. It's too low for the infection because there is no health person in our life. And influenza are staying in our life. So one person can be infected 1.5 person. So we have pandemic for the influenza, and similarly Ebola as well. And if we check the MERS here, the basic reproduction number R0 seems 16. So it means that one person will be infect around 16 person. So there are huge, this number is huge for us. So still for the measles, we have a pandemic, and similarly smallpox as well. And if we check the COVID-19, which are fighting these days for this disease, is around 2.5. So still we have a pandemic, and one person can be infected around 2 or more person. And after giving this information about the basic reproduction ratio, I want you to show how these models are working. So first, I will explain I will take one single strain SIR model, and I will try to explain the structure of the model. Here, we divide the population in 3 compartments, S, I, and R, like chemical-mechanistic. And S here represent the suspectable class, I infected, and R recovery class. And here we have some parameters: lambda recurrent rate, mu is a natural rate, and beta is a transmission coefficient from S to the I, and gamma the transmission coefficient of infected to recovery. So if any suspectable person contact with the infected person, it can be removed that this S class, and it will become to the infected class at the rate of the beta. And any person can be removed from infected class when take its treatment or and recover from the infected class at the rate of the gamma, and it can be removed that class with the natural death rate. It means that we are assuming that. And D here, we forgot to write here. D here, death rate from the disease. So other living part from the infected class is the death. And with using this structure, we construct our system 3x3 system, and we are trying to do the analysis of this one. First of all, we find the equilibrium points for this disease. What is the equilibrium point? When we equalize the 0 to the right hand side of this system, 1.1 here, we can find these 2 equilibrium points. The first equilibrium point, we are saying that this is disease-free equilibrium point, because if you can check here, I part is 0 here. I is infected. So in that area, we don't have any infected person. And the second equilibrium point, we are saying that it is endemic equilibrium, because here, as we can see, that there is infected person. So in that area, individuals can be infected. And we are saying that the basic reproduction number is too important for the studying this kind of compartmental model, and decided if we have disease or not. And the easiest way to find the basic reproduction ratio is the next generation matrix method, which given by Dickman and Hesterberg. And with using their methods, we construct our matrices and we found our basic reproduction number. Now, if we check here the basic reproduction number, how it works, we are saying that we want to decrease it under the 1. So we want to say that it is less than 1 to disease dies out. How we can decrease this R0? Here we say beta is a transmission coefficient of susceptible individual to the infected. So if we decrease the denominator, it means that R0 gonna be decreased. If we increase denominator, it means that R0 will be increased. So transmission coefficient of susceptible individual to the infected individual. So it means that we have an infected person here. So if we decrease the beta, the rate of the beta, it means that R0 value are decreased. So if we decrease the contact of the susceptible person to the infected person, then disease will die out. Or here we say that gamma is a transmission coefficient of the infected individual to recovery. So maybe infected person can take some treatment and they can remove from the recover from the infected disease. So if we increase the treatment, or if we can increase maybe vaccine, then we can remove these classes. It means that if we increase the denominator, then R0 again will be decreased. So one of the decreasing for the R0 value is enough to increase the treatment, or as I told you, maybe vaccine or anything else that infected individual can be recovered. And here, as a mathematically, we can talk about here, natural death rate and death rate from the disease. It mathematically makes sense for us because when we decrease natural death rate, R0 will be decreased as well. But what is the meaning of death, natural death? It means that the person gonna be dead. If it is dead, then it will be not contacted with the infected class, so it will be not infected. So infected class gonna be decreased. Similarly, if any person dies because of the disease, then again they will be not contact with the susceptible person, and they will not infect to any susceptible. Therefore, R0 disease again decrease. So R0 value decrease as well. These make sense. Of course, these situations are not our expecting. And so, with using this kind of decision, we can decide how we can control our disease. That's why R0 value is important for us. And this, I want to give some knowledge about the stability analysis. We are studying the equilibrium point for talking about the stability analysis. Why we need the equilibrium points are to be stable. If they are not stable, then, for example, let us say that infected person is for around 400. And if we say that our endemic equilibrium point is stable, it means that our infected person will be around these numbers. So maybe 390, maybe 410, but it will be around these numbers. So the infected person will be around 400. But if it is not stable, we can't say it. So if it is not stable and we say that the equilibrium point is 400, we can't say that our life, the infected person in our life will be around 400. It can be 600, it can be 1, it can be 2, we can't know it. So to study with the modeling, the stability part is important for us. If we can say that our equilibrium points are stable, then we can talk about, with using this modeling, we can talk about the number of the infected person, number of suspectable person, and something like that. And of course, this here I gave some mathematical definition, and we apply this theorem and definition to our equilibrium point, and we gave two theorems here. The first one, it says that disease-free equilibrium is asymptotically stable when R naught is less than 1, which means that we don't have disease. And the endemic equilibrium A1 is globally asymptotically stable when R naught is greater than 1. And after giving this information, I want to talk about the COVID-19, which is the most important virus in our life nowadays. As we know, we have a vaccine during these days, and we are talking about we have to take vaccination, and we need vaccine for the fighting for this disease. And we want to see this structure. To see this, we construct our compartmental model. We divided the population in a 6 compartment. First compartment is suspectable person. The second, which is vaccinated individuals, and any suspectable person can be goes to the vaccine class at the rate of R. So this R is a vaccinated vaccine rate. And any suspectable person can be contact with the IM. Here, I sub M means that mild infected individual, and IS is a severe infected individual. So any person can be contact with the mild infected individual, and it can be infected with the rate of beta, and any suspectable person can be contact with the severe infected individual at the rate of the beta 2, and it can be disease. So, and mild infected and severe infected person can be removed at a rate of gamma 1 and gamma 2, and unfortunately, this only a severe infected individual can be died at the rate of the day. And here we want to see the effect of the vaccine. So when the vaccinated person contact with the mild infected or severe infected individual, what is the probability that there will be impact? We want to see the effect of vaccine here. With creating this kind of model, we are looking for the answer of what is the effect of the vaccine. With using the model, we create our system, differential equation system, and of course, with using the next generation matrix method, we found our basic reproduction ratios. Here, if we check that, I just explain how the vaccine plays here. So, if we can check vaccinated person, the rate of the vaccine. So, suspectable person, we say that vaccinated at a rate of R. And R is here, if we can see that, on the denominator. So, if we increase the vaccinated person, it means that it decreases our mild infected individual. And similarly, for the severe infected, we can say the same thing as well. And here we have some theorems. And with using these R naught values and our model, we can do our numerical simulation. If we check these simulations here, in the figure 1, we assume that there is no vaccine in our life. If we don't have a vaccine, we have pandemic, and the R naught value is around 3 0.63. So it means that one person can be infect 3 person in our life. And we have death. And similarly, if we check the second figure, still we have we assume that 20% of the individual are vaccinated, and still we have disease here. But if we check the severe infected individual and death rate, it is if we have 20% vaccine in our life, we have disease, but not the previous one. It is not serious as the previous one. And in the third one, we assume that the circulation per person is low from the previous two. And we have 20% vaccinated person. If we check this, still we have mild infected disease. The R0 value is around 2.30. But if we check the death people, it's lower than the previous one. So the vaccine, we can say that vaccine can affect to decrease the death rate for this disease, of course. And if we check the figure 4, we assume that we increase the vaccinated rate, and we assume that there are most people on the circulation. We can see that still we have a mild infectious, but dies almost zero. Not zero, we can't say that it is zero, but it is too low for us. And the last figure says us that if we increase the vaccinated rate and if we decrease the circulation person, we can fight this disease and disease can be died out. Here, I just try to give some information. If we check this table, here we have vaccinated people in the countries, and they take the least one dose for COVID-19. If we check the UK, they take 46% their country. And if we check today the UK, there is no dies during these days because of the COVID-19 in UK. So it shows us vaccine is really important for controlling the disease. And with using our model, we can say the same thing. And I have a lot of time, I know, but just I want to give a bit more information about COVID-19 model for Cyprus. And we create our model, which is 9, we divided the population 9 compartments, and here we have a parameters, and with using this, we found our basic reproduction ratio, which is a bit huge. And with using this, until during this, in the beginning of the disease and at the end of today, we are calculate R0 values. And nowadays, the R0 values for the North Cyprus is around 1.9. So still we have a pandemic in our country. And this graph shows the first wave COVID-19 in our country. We say that around 140 persons will be infected. And we take our control, we lock down, and we obeying the rules, and we fight these illnesses. In the I'm talking about the first wave. Then after, of course, we have to open under the controlling, and after we open, we have the second wave COVID-19 in our country. And here we draw our graphs for today. If we check here, this shows today for our life, and we can say that infected person which taken treatment in the quarantine and infected person which they take their treatment in the hospital, and we draw our graphs. We can see that during these days we have the same infected individuals in our country. And here we want to see the asymptotic individuals in our life. If we check the gray part, we can see that we have asymptotical infected person in our life as well. And this is the last model which I want to give an explanation. We say that we have a B117 variant, so the COVID-19 have a mutation. So we construct our model for the variant B117, and we can see from here, using this model, we can see from here the B117 variant person, infected person, will be around 400, and before B117 COVID-19 gonna be fell down, and here you can see the R0 values. And from here, I just want to tell you one more thing. We are expecting that if we don't change any condition in our life, so still we have a quarantine, if we obeying the rules, which I mean that using mask or something like that, we are expecting that the infected individual will be around 400%. But if we change our conditions, we can't talk about for anything. But if we don't change, we are expecting that infected person will be around 400%. And these are that I want to explain to you. Thank you for listening to me.
Thank you so much, Dr. Kaymakamzade. Dear participants, we will now hear from Dr. Selin Deliceırmak, who will present a project titled Understanding Climate Change in Meta-Omics Era. Dr. Selin.
I have a connection problem. I guess I couldn't hear if Ranim introduced me, but
yes, I did.
Realized that I am online now. So sorry for that. Thank you, Ranim. I'm really excited today to be able to introduce my research topic, marine microbial ecology, under the presentation name of Understanding Climate Change in the Meta-Omics Era. So today, throughout my talk, I would like to first introduce some facts about climate change, and then I will move on to the importance of oceans and marine microorganisms and how we can relate them with climate change, and then what we can do as a molecular biologist to understand the climate change using the omic studies. And then I will shortly talk about ecosystem models. And here I would like to thank to Dr. Kaymakamzade that she already introduced the basic principles of mathematical models. And then I would like to also continue with our role. Again, I would like to stress how we can integrate the data we got from molecular studies into ecosystem models. And then I would like to end my talk explaining what we are doing in our universities as a marine studies. So let's begin with some facts about climate change. Intergovernmental panel on climate change actually defines the climate change as a change in the state of the climate that can be identified by change in the mean or the variability of its properties. And the important point is that this should be persisted for an extended period of time. This is typically decades or longer. So we are not talking about day-to-day change. It's a weather, it's not a climate change. So if we are talking about a climate change, we need to see change for an extended period of time. And when we are talking about climate change, you might have heard that most of us talking about also increase in the carbon dioxide concentrations in the atmosphere. And studies already demonstrate that carbon dioxide concentrations are currently higher than they have been at any point in human history. So global temperatures are rising at an unprecedented rates. So here we see a graph which is actually explaining us from 1750s till now how atmospheric carbon dioxide concentrations are changing, and how here with the purple color, and how the carbon dioxide emissions are increasing. So from this relationship, you may already guess that the main cause of the carbon dioxide increase in the atmosphere is due to carbon dioxide emissions. And when I was presenting or defending my master thesis during 2013, the carbon dioxide concentrations in the atmosphere was almost 400 ppm, and as you see, we already passed that number. We already passed it, and we have more than we have approximately 4010 ppm carbon dioxide in the atmosphere. The last time the atmospheric carbon dioxide concentrations were this high was actually more than 3 million years ago. It corresponds to time when temperature was 2 to 3 degrees higher, and the sea level was 15 to 25 meters higher than today. So recently, it has been debated that if this climate change is due to anthropogenic forces, meaning the carbon dioxide emissions that humans cause, or it's due to the natural fluctuations in the climate system. So what they were asking is, if Earth faced such high concentrations earlier, then what is the current problem? So I'm going to give a pause here and let you think a little bit about it. What might be the current problem if we already faced such high concentrations early in the Earth history? The reason is actually the rate of increase. We afraid that it increases such a high rate. So the annual rate of increase in atmospheric carbon dioxide over the past 60 years is about 100 times faster than previous natural increases. So after introducing the basic facts about climate change, let me come to the point and let me explain why oceans are important. Why do we need to understand the dynamics of oceans? So world ocean actually provides us clean air, climate regulation, transportation, recreation, economy, food, medicine. But here I would like to talk about specifically the clean air and its carbon dioxide storage capacity. So it might be surprising for most of you, but the half of the oxygen that we breathe is coming from the oceans. It's produced by the photosynthetic microorganisms. And the other important thing that is important for ocean is they store 50 times more carbon dioxide than our atmosphere. So this is basically what I would like to concentrate on. How oceans store carbon dioxide. If ocean stores carbon dioxide, it might help to reduce the carbon dioxide in the atmosphere and reduce the greenhouse gas effect. So here you see here in the right-hand side, you see a sketch of a marine environment. And here we have two important mechanisms to store carbon dioxide in the oceans. The first mechanism is biological carbon pump, and the other one is microbial carbon pump. So biological carbon pump is simply the phytoplankton, meaning photosynthetic microorganisms. They fix carbon dioxide, inorganic carbon dioxide in their body as an organic carbon. And then when they're sinking down throughout the ocean depths, they carry these organic carbon to the sediment of the ocean. The second mechanism is mediated by mostly heterotrophic bacteria, and this is my study topic. I'm trying to understand their dynamics. So what they are doing basically is they use this organic carbon and convert it, produce some inorganic nutrients, and also they store this organic carbon long-term in the sediment. So marine microorganisms are unseen majority. So they are ubiquitous in all marine ecosystems, and they have huge impact on marine biogeochemistry. So let's have a look at their role. So they they are responsible for remineralization of organic matter, and they provide nutrients for primary producers here with these loops. And microbes are also responsible for the degradation of organic matter in the ocean. And this is the key for maintaining the balance between produced and fixed carbon dioxide. They export carbon to the depths of ocean, and they maintain nitrogen and phosphorus cycle. So what, as a molecular biology and genetic background, what we can do, how we can help to understand climate change, or how we can introduce these information we gain from omics studies into ecosystem models. So before I begin, I would like to have a short summary here. So the ocean and coast provide critical ecosystem services such as carbon storage, oxygen generation, food, and income generation. And marine microbes are responsible for the degradation of organic matter and key for maintaining the balance between produced and fixed carbon dioxide. So they help to decrease the atmospheric carbon dioxide, which causes greenhouse gas effect. So even if we know that they are important for the ecosystem, marine ecosystem, and world's health, they are the least known compartment of the ecosystem. It's basically because it's really difficult to sample them because they are having a really complex and dynamic habitat. We don't have a suitable microbial sensor. Sampling is hard. So this is actually what you see here in the picture is my green sensor. Maybe 20 years later, I might have one, but we don't have such sensor that we can sample and then gain the data from them in a short time. And the other important difficulty is that we don't have sufficient number of relevant culturable model organisms. So you might have heard that microbiologists are culturing Escherichia coli or other organisms. But if you take a water from the sea and try to culture it, 90% of organisms you will lose. You cannot culture them. So we don't have enough number of model organisms to culture. So I would like to introduce two important techniques that we use in order to understand their dynamics. So the one of them is metagenomics. The other one is metatranscriptomics. So after the invention of next generation sequencing, the developed methods of metagenomics and metatranscriptomics allow a detailed study of bacterial communities without needing to culture them. This is really important. With these techniques, we don't really need to culture them. So we're not losing 90% of organisms. Using these methods, actually, you can study community structure, their relative abundance, as well as their active metabolism. So metagenomics answers the question, who is there? Who is living there, out there? And metatranscriptomics answers the question, what they are doing. So these omics studies provide unprecedented amount of data, specifically about the evolutionary relationships and the functional trait of microbial communities that regulate global biogeochemical cycles. Here I would like to shortly give information about these two techniques. So if we have a look at metagenomics, it answers who is their question again. You can simply use 16S rRNA gene and amplify it in order to have the information. And this 16S rRNA gene has highly conserved regions between bacterial and archaeal species. And then it constitutes highly variable regions that makes taxonomic studies possible. Also, 16S rRNA gene has wider amount of library. So you can use your data and compare it with the data in the database. Metatranscriptomics is basically answering what are they doing there. And it's based on studying mRNA in the environmental sample. So you gain the information about metabolism and you can answer how they respond to the perturbations, long-term or short-term. So here I would like to introduce ecosystem modeling approach. The ecosystem models are actually not different than the mathematical that Dr. Kaymakamzade introduced us in the previous talk. So they are the abstract representation of an ecological system. You have a prey and a predator, for instance, and using mathematical equations, you try to explain how many species are eaten by a predator in a given period of time. How fast and how many of them were eaten. But since the microbes are dominant components of the marine ecosystem by abundance and by their metabolism, so these traditional approaches where you represent them by their only abundance, so giving roughly their number, how many bacteria there is a really basic approach to understand their complex dynamic system. So the tradition from nowadays, the studies are concentrating more on gene-centric approaches. So gene-centric approach is basically where you can integrate the information you gain from Meta-Omics studies. So the abundance of a specific gene, or the abundance of a specific, the abundance of the organisms who carry these genes. Okay, it's basically refers, this gene-centric approach basically refers that you use genetic data as an input in your model to predict what's going on in the ecosystem. So here I would like to stress some important points that I have mentioned until now. Microbial communities are ubiquitous throughout the oceans and exert great influence over the ocean chemistry. They are the least known compartment of marine ecosystems, and they are rarely included explicitly in models of marine biogeochemistry, such as that used to predict the response of oceans to environmental problems, such as climate change. And modern molecular tools provide an invaluable window into the marine microbial world by identifying organisms and their metabolism through the analysis of genetic material. So what we are doing in our universities, marine studies has started with the 1001 project, and it was a collaboration of nearest university, University of Kayrinya and Middle East Technical University, led by Prof.Dr. İlkay Salihoğlu, and you may remember him that he gave a talk yesterday in the morning. The topic of the project was the determination of influence of anthropogenic processes on the Silesian Basin marine ecosystem. So this was the project that actually marine studies start to develop in our country. The aim of the study was to investigate the area, and just note that there was a few research has been conducted in that area. So to investigate the area for the effect on ecosystem of anthropogenic and natural change. So if you have such aim, you require a long time observation. So under the framework of this study, 26-month-long cruises were organized, and the Kıbrıs time series was initiated. Here in this map, you see where the location of Kıbrıs time series. So from the start of 2015, we are having monthly cruises and we have a huge data on physical, chemical, and biological properties of oceans. And here, before I end my talk, I would like to stress that we already have DNA samples to be studied in order to understand heterotrophic bacterial dynamics and the effect of anthropogenic forces. And we have long-term data on DNA samples from 2015 to 2018. So thank you for your patience. It was a great pleasure for me to have this opportunity, and I would like to thank to our students from Genetic Student Association. This is all I would like to say. If you have questions, I'm ready to answer.
Thank you so much, Dr. Deliceırmak. Dear participants, please welcome Associate Professor Dr. Sezgin, who will give his presentation titled Genome, Microbiome, Metabolome, Who's Controlling Whom? I'd like to handle it over to you, Professor Sezgin.
Okay, thank you very much. Can you hear my voice?
Yes, we do.
Okay, so let me try to get my computer going. Share screen, application. Can you see that now? Sorry, let me go back. This is all right. Can I start now? Can you just tell me if I can start? Can you see my screen? Okay. Okay, I hope you can see my screen right now. Well, thank you very much for your invitation. So hopefully this talk will follow nicely after Professor Selin's talk. Microbiota, as you may know, is a quite popular topic, and it's when we say microbiota, it means that it's the totality of all microscopic living forms on us. It's let them be, most of we talk about bacteria, but we should not forget about the archaea, fungi, and viruses as well. And the importance is that because cumulatively there are more cells and more genomes that are on us, living with us, compared to our own cells and our single genome. And we know that there are several positive effects of microbiota, for example, such as helping the digestion, synthesis of certain vitamins, epithelial homeostasis, detoxification, reduction of inflammation, and so forth. Okay, many positive benefits of healthy microbiota. And moreover, disruptions in the microbiota has also been associated with a number of metabolic and physiologic diseases. For example, obesity, diabetes, cardiovascular diseases, allergies, okay, inflammatory, autoinflammatory disease conditions, and so forth. And even psychiatric disorders has been linked to problems and dysbiosis in our microbiota. Okay, and of the microbiome that we have on our bodies, the gut microbiome is the most richest in terms of the number of taxa and the abundance of taxa. Okay, so we will the gut microbiome will be the focus of my talk today. As you can see, the number of publications within the last 10 years has increased logarithmically, drastically, okay, both for microbiota and regarding microbiota and metabolism. So if you do a keyword search in PubMed, you are going to see these results. Why is that so? Just like Professor Selin told you about this, this is because since the last 10-15 years, culture-free, culture-independent identification methods has become quite popular. Just like she told us, we use the next generation sequencing. We just get a sample. We don't know what's in it. We don't need to know what it is in it, and we don't need to optimize any cultural conditions. We just take them and do genetic analysis on the 16S region. We sequence with next-gen sequencing, compare it to certain databases, these genetic data, and identify the taxon composition. This is revolutionized really the microbiome research. Okay, and given this amount, given this revolution using this next-gen sequencing and all these experimental approaches, a number of factors has been identified that seems to be influencing the microbiota composition, such as the major ones are like infections, as you can imagine. You can get a diarrhea, for example, from food poison, you can think about that. Clearly drugs, we call them as xenobiotics. For example, antibiotics can alter your microbiota. But also lifestyle, diet, and sex also have an influence on them, okay? But how about genetics, okay? Looking at the effects of these non-genetic factors, we can call them, is easier. But looking at the genetics, effect of genetics on microbiota composition has always been challenging. So a UK study, one of the primers on that subject, they looked at the twins, okay? They do the twin study. If a trait from a quantitative genetics perspective, if a trait is heritable, you expect the relatives to be much more similar to each other compared to non-relatives. For example, so what they did is that they took monozygotic twins, because they are supposed to be nearly 100% identical genetically, and then dizygotic twins, which are at least around like 50% identical in terms of their genome, and unrelated individuals, okay? And what they observe is that monozygotic twins, the microbiota of monozygotic twins are much more closely related to each other compared to the dizygotic twins, and which are compared to the unrelated individuals. As you can see, they are there is statistically significant there because, but you can see there's a huge variation, and the effect is rather tiny. Okay, so clearly even among the monozygotic twins, so this at least tells us that it's heritable traits, because the more genetically related you are, the more similar your microbiota is. Okay, and when they looked at it, so what is heritable? Because microbiota is a strange concept. We are looking at thousands of taxa in an environment. You can think about this, but here we are talking about the gut environment. And when they looked at this taxa, their taxonomic relationship, they did a phylogenetic tree, and they colored the effect of the, let's say, heritability of each one of these taxa. So, for example, this is the p-value, means the statistical meaningless. The more red it is, the means that it's more heritable. So when you look at the overall distribution of taxa that are being identified in this study, most of them are like blue, light blue color, meaning that it's really not heritable. So the similarity between the monozygotic twins or the unrelated ones has not been all that different. But what was heritable was that it was only the very tiny amount of these redly colored branches of the tree. So it's only very specific taxa has been seems to be heritable, rather than an overall taxon distribution. It's only seems to be pertinent to certain specific taxa. And they identified, for example, that to be, for example, the Crystallinacea. Okay, so if you're among the monozygotic twins, so this is the regression line, you can think so. So they are more highly correlated. So if one of the twins has a higher amount of Crystallinacea, the other monozygotic twin has also a higher level. So this relationship seems to be degrading when you look at the dizygotic twins and the unrelated individuals. Okay, so this was the first clue that microbiota can be heritable. But the questions are still rising. So which genes are these? So how can we find these genes that seems to be involved in the microbiota composition? So again, in order to be able to look at that, we are going to go into the quantitative genetics. So these are the Galton and Fisher, maybe from your genetics classes, you may know. They are like the fathers of, let's say, quantitative genetics. and the formulations, and also a number of important topics in statistics. Okay, so for any complex phenotype or any type of phenotype, we can partition that into genetic and non-genetic, such as environmental factors and its sort of interaction. Okay, so this can be anything: your height, weight, BMI, blood pressure, obesity, cardiovascular disease rate, cancer risk, whatever you want to put in as a phenotype, you can model it as such. Okay. For example, here, when you take a group of individuals, order them with respect to their height, in most biological actual phenotypes, you are going to get this similar, a normal distribution. Okay, so and then you can partition them into this a genetic component, which is a genetic matrix. Okay, you can measure only one gene that's maybe an interest to you. You can call it as a candidate gene, and a number of environmental factors. Okay, this is how we formulate it in terms of quantitative genetics, and how this is how we do, for example, genetic studies to identify genetics of not only diseases but any type of complex phenotype as such. You can see in this example the height. So my question is that, can we also model microbiota in a similar way and identify the genetic factors that's contributing to this microbiota composition? Okay, and so far the studies, at least for example in this UK twin study, it suggested that the genetics has a much tiny effect compared to these environmental effects, okay? Such as diet, lifestyle, age, and sex of the individuals. Okay, so is that really the case? Okay, is this true? A follow-up study looked at that because environmental factors seems to be highly confounding these microbiota compositions, trying to incorporate genetics and environment and using humans especially has been quite challenging. And this is one of the unique studies that looked at a specific population. It's called the Hutterite population. These are a very unique group of individuals that are living in a community together. They have very similar diet and lifestyle. Say they looked at they looked at their microbiota composition. Okay, measured it with these molecular next-generation sequencing techniques. They also genotyped these individuals, and they do the genome-wide association study. GWAS stands for a genome-wide association study, meaning that you looked at hundreds to thousands of different kind of markers, genetic variants, in a genome of an individual, and you look for associations, statistical associations, and you end up with this plot. These are called Manhattan plots. And on the x-axis, you see the 22 autosomes of the humans, and each one of these dots is a result of a statistical test result. It means that whether this, for example, this variant has anything related to the microbiota composition or not, whether this variant has anything, any relationship with this microbiota composition or not. Okay. And they sort of, and because we are doing so many tests, so many statistical tests, in order to reduce the false positive, or in statistical terms, type 1 error rate, you accept only a certain p level to be significant. For example, here, at least you need to have at least 10 to minus 5 or 10 to minus 6, means that 1 in 100,000, 1 in a million chance to observe this statistical result by chance, so that you accepted something biologically meaningful. And when they looked at it, they only identified one genetic variation, barely sort of actually statistically significant with the stringent criteria, and they identified it to be sort of related with the Akkermansia, genus Akkermansia, a group of bacteria levels. As you can see here is the individual genetic test results. So if you have a CC type of a genotype, your Akkermansia level seems to be lower compared to the TC genotype, for example. Okay, so this was the pretty much the only study that was able to find barely statistically significantly any effect. Right, and it still suggested that maybe the genetic factors contributing to the microbiota can be little compared to the environmental factors, but they are not zero. We should keep that in mind. So this comes to, so clearly we cannot do these type of studies which identify, at least in terms of the genes and their genetic variants, just using the humans. So there's a need for a model organism. So model organisms, as you know, you can do experimentation on them. There is quite extensive genetic information and phenotypic information for them. That's why they are called model organisms. And Drosophila melanogaster, I'm sure you must have heard from your genetics classes, biology classes, is a model organism that's been, or at least a model organism for over 100 years, has been used in all types of genetic fields. Molecular genetics, quantitative genetics, neurogenetics, developmental genetics, you name it. It's always been, there's always some group of researchers that's using Drosophila as a model organisms. Why? Because at least 75% of the genes that we see in this fruit fly Drosophila is homologous with humans. So you can translate the genetic findings from this Drosophila to humans rather easily. There are also this fly also has analogous organs. There's a heart, there is circulation, is liver, kidney, is the central nervous system, muscular system, okay? And more importantly for us, it has a digestive system, okay? So when you look at compare the humans and the Drosophila, the fruit fly, they're quite analogous organs. Starts with feeding, starts with mouth, and there is a stomach where the digestion occurs, and there is the colon, like the digestive systems, like our intestines, where the absorption of the nutrients, continuation of the digestion, absorption of the nutrients, and the excretion happens at the rectum. Okay, so it's quite analogous actually to our own. And when you look at the actual cellular level, you can also see that there's an epithelial layer that's involved in the digestion and absorption. But in humans, obviously, it's a slightly a little more complicated. There are a lot more immune cells involved, and the mucus layer is thicker, a little more complicated biochemically compared to the Drosophila. But clearly there is, again, they are rather similar. It's just a simpler form, but overall what is happening seems to be quite similar between humans and Drosophila as well. And moreover, people has been doing quite molecular studies and microbiological studies showing that this mucus layer is also, just like in humans, rather important in terms of the interaction between bacteria, let's say in the Drosophila, and the host interaction. Okay, so then does Drosophila melanogaster have a microbiota? And people looked at it in the laboratory strains. These are not like wild caught animals. These, for example, this W1118 or Canton S, famously used, experimentally used fly lines that has been made in the lab for hundreds of generations, and they have a unique microbiota themselves. And there is, again, you can see the male, female, there's a sex difference in them as well. Okay, so we have these lactobacillus, Assoc, enterococcus, and so forth. It's a diverse, simpler but still diverse microbiota. And even this microbiota, just like in humans, there's a sex difference, and also by age the microbiota changes. So it is an aging effect. So this shows that Drosophila can be a good model to study microbiota. So this is what I am going to focus on as well. And moreover, because Drosophila is a model organism, a number of tools has been developed, not only molecular tools, but at the organism level. At least around like 5 years ago, a number of special Drosophila lines has been developed. These are called Drosophila genetic reference panel. So these are around 200 inbred lines, means that their genomes they've been inbred such a level that every individual in that specific line, for example, inbred line number 1 is identical with respect to each other. Obviously, the only difference is the sex chromosome between males and females. Okay, so they have 200 of these lines, okay, representing 200 genomes. All right. Moreover, their genomes has been sequenced and it's available. And you can also purchase, you can buy these lines from the Drosophila stock centers. And so far, they've been widely used in order to understand the genetics of complex phenotypes. There's over 30 publications using these DGRP lines, the Drosophila genetic reference panel lines, to answer the genetic basis of complex phenotypes. Okay. And okay, then, so does DGRP lines have a microbiota? People have started using these lines in terms of bacterial microbiota, maybe I should say, to be more specific. Yes, indeed. Even these laboratory made inbred lines also have a, let's say, a microbiota of their own. All right. So what I'm going to talk here is that slightly different. We are going to talk about microbiota, but I am going to focus on fungal microbiota. All right. Why fungal microbiota? So if you look at, again, in the PubMed, you do a keyword, for example, with microbiota, within only the last 10 years, you are going to get nearly 72,000 results, okay? But when you say fungal microbiota is a keyword, you get less than 3,000 results. So as you can see that it's much less studied, it's much less understood microbiota. It's also called like a neglected microbiota. Unlike the bacteria, these are eukaryotes we are talking about, because so their genomes are larger, a lot more genes are involved, so potentially a lot more interactions with the host. So we treat that, it should be interesting. You may be familiar with actually our fungal microbiota if you get fungal infections. Okay, you can get some fungal infections on your hands, on your foot, and so forth. And you know, if they are a lot more harder to get rid of compared to bacterial infections. And actually, there are probiotics you can buy. For example, if you have diarrhea, whether due to food poisoning or some infectious agents, for example, traveler's diarrhea, you can take this Saccharomyces boulardii, which is just like a yeast strain, just like very close related to Saccharomyces cerevisiae, and it's very effective in terms of controlling your diarrhea. Clearly, this tells us that fungal microbiota should have a role in the dynamics of our own gut microbiota as well, okay? So my question was this, okay, then Drosophila have a microbiome. So the fungal microbiota is also called microbiome, okay? So I may use both words interchangeably. Okay, so, and then do host genetic factors, just like the bacterial microbiota, affect this microbiome? And does the microbiome influence host metabolism? So in this aims of the study was that because it's much less studied, we don't know. We didn't know that whether these DGRP lines have a microbiota of themselves. So first we aim to look if DGRP lines have a microbiome. Then we wanted to quantify the major fungal taxa in all of these DGRP lines. Do a genome-wide association study because the genomes of these flies are available, these DGRP lines, and identify if any host genetic factors influences the microbiome. And then we measured also the metabolite levels in order to see whether any of the metabolite pools correlate with any of the major fungal taxa, okay? So we raised these flies, these inbred DGRP lines, 120 of them under identical conditions, under a number of generations. Then we extracted here, as you can see, the digestive system. We did DNA isolation. We do PCR, the ITS region, just like now in the next slide, I'm going to show you a little more about it. Like 16S is for bacteria, just like Professor Sinem told you about, Sibel told you about. We have the ITS region for fungal microbiomes. And then we did next-gen sequencing and identified the fungal taxa. So this is the ITS region, just like in the 16S region. So this is still a ribosomal region, important for eukaryotes. And very similar, it's like a nice genetic biomarker to identify the taxa. There is a lot more data on them, so we can use them, just like the 16S. We have quite a bit of these databases. We can get this data and compare our results with these already established databases and identify taxa, okay? So when we looked at it, we only, because we are short on money, we were only looking at around like 10 DGRP lines. But we have observed that there is a microbiome, or let's say fungal microbiota in these DGRP lines. Ascomycota, Basidiomycota, and Zygomycota were the major taxa, okay? And then, because we don't have enough money to look at all 120 of the DGRP lines, to use next-gen sequencing, which is rather expensive, we identified, we focused the major Ascomycota and Basidiomycota as the taxonomic level. We keep in mind that these are not species, they're all the way up the phylum level, okay? Using our own sequence data, we designed specific primers for Ascomycota, Basidiomycota, and we did qPCR because we want to get a quantitative estimate about how much is Ascomycota, how much of the Basidiomycota, and how much of the Zygomycota is present in these DGRP lines guts, okay? And we were successful in doing this Ascomycota and Basidiomycota, okay? Because Zygomycota has turned out to be rather complex to assay quantitatively, okay? So these are the qPCR results. And we also did these metabolite assays, such as we measured protein, glucose, trehalose, which is an insect blood sugar, glycogen, and triglyceride metabolites. Glycogen is a major storage metabolite in the muscles, just like similar to us in these fruit flies. And the fat tissue is also a triglycerides are representing the fat metabolism. This is the next important metabolite pool. Okay, so when we look at the results, indeed, in these DGRP lines, in 120 of them, there is a distribution of Ascomycota. Some of the lines has very little, some of the lines have quite a lot of Ascomycota, the fungal taxa inside them. So is the case for Basidiomycota. Okay, there's a range of them, which is quite nice and interesting to do. And so the ratios is also is changing. Okay, so there is clearly a diversity of fungal microbiota in these DGRP lines. All right. And in terms of metabolites as well, as you can see, there are these fruit flies with, for example, large amounts of glycogen, and there is also the ones which are large amounts of fat and so forth. Okay, there's a nice distribution for all of these metabolites. So if you summarize them actually cumulatively in terms of a metabolite, so imagine that we are taking a metabolite profile picture of these 120 lines, and overall you can see that the big metabolite pools, such as the especially the glycogen and the triglycerides, are the major determinants of these pools, because the trehalose and glucose fluctuate quite a lot, but the others are stable because we did these measurements obviously multiple times. So there are flies, these lines, that are, for example, high fat content and lower, let's say, glycogen content. Okay, and there are also flies that have a higher glycogen content, so they are storing their energy in the glycogen form in the muscles, compared to the fewer fat content. Okay, so this shows the metabolic diversity in these lines as well. All right, so then we did this genome-wide association study. What does it mean? Because we have the genome information, genomes of these fruit flies has been sequenced. We can get them from the servers. We download them, and then you can do genetic association study. This is over 1 million variants. So again, these are the individual results. So in the y-axis, you see the p-value of it. So anything above this blue line means that biologically meaningful, statistically significant, and meaning that there is maybe a biological effect. So we identified host genetic factors that can be contributed to the Ascomycota amount, Basidiomycota amount, and their ratios. Okay. And indeed, we were able to identify some of these genes. I will get into more detail later on. So for Ascomycota, there are a number of genes. So Basidiomycota, the number of the genes. So these were the first time we have observed them, actually, even in the literature. And these are independent, obviously, genome-wide association studies. And it's good that we are coming across with the same, we are able to find the same genes, even using, for example, their ratios and so forth. Okay, so this, we think that they are biologically meaningful results. And when we looked at whether any of the taxon here has anything to do with these metabolites, so has any effect on the host metabolism, we observed that Basidiomycota amount and the triglyceride amount show the correlation. So, and none of the other correlations were statistically significant, but this showed a statistically significant correlation. So this suggested that Basidiomycota may be something to do with the fat metabolism in these fruit flies. Okay. And because because we have all these tools, Drosophila is a model organism, and we have all these tools and measurements with us, let's measure, let's do a genome-wide association study and take a look at if there are genes on the host that are also contributing to these triglyceride levels, and see if these genes that are contributing to the Basidiomycota level and the triglycerides are similar, different, or is there any overlap? Because if you remember, Basidiomycota seems to be influencing the fat metabolism. We did this genome-wide association study and identified a number of genes. And so in this table, actually, we are looking at the genes that are seems to be influencing Basidiomycota amount and the triglyceride level. So important thing is that none of them are overlapping. Okay. And I'm only showing the genes that are also human orthologs. As you can see, all of these important genes have human orthologs. So our results has a meaningful in terms of humans as well. And their function, there is a number of functions. And we also check the databases in terms of the Drosophila gene expression, and they are actually in meaningful tissues in terms of their gene expressions. Okay. So in the conclusion, at least this short conclusion, what I told you so far is that so the Drosophila, even in these inbred DGRP lines, seems to be a rich fungal microbiota. So, and as you can see, these DGRP lines can be a good model system to study host fungal microbiota interactions. So we have seen that host genetics can influence fungal microbiota. Host genetics can also influence fat metabolism. And seems to be that, and then the fungal microbiota, especially the Basidiomycota, can influence the fat metabolism as well. And as you can see, under the genetics, fungal microbiota metabolism interaction seems to be true different and independent genes. Okay, so suggesting that there are different biological pathways are involved in this. So this is also another level of analysis that you can take. You can do gene protein interaction network analysis. So this is an example from the string let's say bioinformatic tool. Okay, so these are all the genes that we identified that are related involved in the Basidiomycota in the list that I showed in a tabular format. So these are the genes that are also influence the Basidiomycota levels and the triglyceride levels, all put into this network analysis tool. Okay, so this network analysis tool searches if there is any potential interaction between them, gene, protein, gene, protein, and so forth interactions. Okay, and it doesn't find anything. So as you can see again, see that none of them are interacting. One of the reasons is that because nearly all of these genes has not been extensively studied, and most probably there isn't enough information about them. Okay. But if you ask this algorithm, okay, this network algorithm, okay, include a few more other genes in order to get some network structure, okay? Are there other genes, if you include them into the network, do you get any pattern? Is there anything? So there seems to be something coming up, okay? Not with these genes are not directly interacting with each other, but if you start including other genes in the same biological pathways that they are in chosen by this algorithm, so you start to get some sort of interactions happening. Okay, so again, these are promising results and some results that needs to be followed upon. And so when you do this same thing with humans, orthologs, if I show you on the table, is that so the Drosophila genes, all of these genes has human orthologs, and there's a lot more data and information available with the humans. So we did the same analysis as if our results were coming from humans. Okay, and you can see by themselves again there is no interaction. But if you ask the network algorithm to include some other meaningful genes, so to see if there is an interaction, as you can see, there's a big, very important interaction networks coming up, and all of them are very heavily studied related to the fat metabolism. Okay, so again, these are very sort of interesting results for us, that suggesting that our results seems to be biologically meaningful. Okay, they are not just some statistical artifacts that we are finding. All right, so genome, microbiome, and metabolome. I think I bombarded you with a lot of information. So who is controlling whom? All right. So the overall sort of dogma or looking at in the literature is that, so we have this genome, we have this microbiome, and we call this environment as an exposome. You may hear about this, influencing the metabolome, our metabolism. Okay, for example, think about this obesity and diabetes and so forth. Okay, all of them seems to be influencing. Or if you're interested in terms of a microbiome perspective, user research is focused on microbiome. People say that they do some genetic studies. They show that microbiome is influenced by genetics. Environment clearly influences, and metabolome clearly influences that. Okay, these are all one-by-one interactions. But what I'm suggesting, these are really insufficient models, because our results clearly show that, for example, we kept the environment constant here, but clearly it's going to have an effect. So the genetics, so the genome, as shown you, has an influence on, at least on the metabolism, at least on the fat metabolism. And genome also influences microbiome. As you can see, it influences Ascomycota and Basidiomycota levels. And microbiome also influences the metabolome, at least the fat metabolism, isn't it? So there seems to be like this network of interactions. And clearly from other studies, we know that exposome is also, it can potentially influence all of them. Okay, so I think this will be a better approach, but it's quite hard to test, especially in humans. But we can use Drosophila, model organisms such as Drosophila, to test whether this model is, for example, sounds. Okay, and using this model, we can identify again novel genetic variants and their effects on genome, microbiome, and metabolome interactions. Okay, so I would like to thank my group. I am in Izmir Institute of Technology, and this is in collaboration with Professor Ergi from Hacettepe University, where they maintain all these DGRP lines. And I would like to end up with an announcement that on April 24th through 28th, we have an Aegean Seminars for Computational Ecology and Evolution. Please search that in Google, and please register and attend a number of, I'm sure, interesting talks you are going to be able to hear all week long. Okay, and I want to end with this note. I think I am going to now exit from my presentation mode, and let me come here. What should I do right now? Are there any questions? Because I cannot seem to have any.
Thank you so much, Professor Sezgin. Yes, we do have 2 questions, if you please. First
Sure,
one,
okay.
About mice as a model organism.
Yes, mouse is also a very nice model organism. It's a mammalian organism. Obviously, a lot of people use that. Their findings quite good. But what you cannot do is that, or it's going to be very, very expensive, is that try to find 200 inbred mice, okay, and sequence their genomes and make them available to that. So you can do all of these things that I told you with that we have done in Drosophila on these mice, but it will be extremely expensive. Okay, maybe days will become, it will be done. But mice is especially used in order to look at a lot of environmental factors, okay, that's affecting the microbiome, and it's been a very instrumental, let's say, model organism to identify again the effects of both sort of some genetic factors and especially non-genetic factors.
Thank you, Doctor. Second question was there a control group in the research conducted to avert the effect of other factors on the amount of metabolites?
Yes. So BR coined the lines, each line is a control group, you can imagine, with respect to the other ones. We take these lines, 120 of them, and we keep them under same, in Hacettepe University, there's an insectivarium, under the same biochemically controlled nutritional conditions, temperature, humidity, daylight conditions, okay? So the environment we try to keep as stable as possible, okay? So in that sense, and we repeat the experiments to make sure that we are getting the same results. So because this is not we have not given any, for example, outside influence. So what this studies, it's only characterizing what is happening, the dynamics by itself. So the next step can be is that you can choose some of these lines as a control group and apply a treatment. For example, do an environmental factor, or you can silence certain genes and look at the effect. Then there will be a control group, and then your experimental group, which you can compare. In this study, there was no treatment group because we didn't do any treatments. This is pure exploration. What is happening in these lines in terms of microbiome and its interaction with the host genetic and host metabolism.
Please accept our deepest thanks, Professor.
Sure.
Thank you.
Sure, you're welcome. Thanks for your invitation. So should I stop my, and should I leave the studio now?
Yes, please.
Okay.
Dear participants, it's my pleasure to now introduce Dr. Elanur Yılmaz, who will present his presentation titled Zebrafish, a Model Organism in the Genomic Era.
Hello, can you hear me clearly?
Elanur, yes.
Yes, okay. Let me share. And first of all, hello everyone. Before I start, I want to thank Selin Deliceırmak and the organization committee for giving me the opportunity to meet you and to talk about zebrafish as a model organism in the genomic era in this such a good organization. The content of my presentation will begin with the description of model organisms that you heard a lot of times today also, and we'll end with the, some examples of the zebrafish research. So let's start with the very beginning. In medicine, animals have been used constantly throughout the history of biomedical research. Scientists who were also interested in medicine, such as Aristotle and Erasistratus, carried out live animal experiments. Galen conducted animal experiments to further his knowledge in anatomy, physiology, pathology, and also pharmacology. And Ibn Züyr proposed animal experiments as a test method to test surgical methods before applying to humans. And from now on, and from these before Christ to now on, by the end of 20th century, model organisms became one of the centerpieces of biomedical research. The historical origin of the term model organism are difficult to trace, but it is clear that the concept of model organism took a firm hold in the 1960s and 70s, in part due to the rise of techniques of molecular biology. So what is a model organism then? Model organisms are non-human species that are used in the laboratory to help scientists understand biological processes. Model organisms are usually the organisms that are easy to maintain in the laboratory facility and affordable and have particular experimental advantages. Their advantages rely on reduced costs, easy maintenance in laboratory settings, affordable but rigorous manipulation tools. So in general, the simpler the organism we use as a model organism, the greater these advantages. The short lifespan of simple organisms and is another key advantages of model systems, which allows both parallel and serial experiments to be performed in reasonable time. And in addition, rigorous genetic experiments can be performed in simple eukaryotes using the powerful possibilities of modern molecular genetics at a relatively low cost. For example, they may have particularly robust embryos that are easily studied and manipulated in the lab. This is useful for scientists who study development, or they may occupy a pivotal position in the evolutionary tree, and this is useful for scientists studying evolution. The experience of many disasters caused by medicines in the history of medicine has revealed the tragic situations that untested substances in animals can cause in humans, and showed the importance of animal experiments in preventing these tragedies. So when, besides these kind of similarities between different model organisms, when scientists look at the genes, they found that about 98% of human genes have a matching version in chimpanzees and 90% in cats. And about 80% of human genes have a match in cows and also in mice. And even some more distant species can share a large percentages of gene with humans. For example, chickens actually share about 60% of their genome with the human, and even bananas share about 50% of their genes with humans, thanks to the evolution. Although a very similar DNA sequence doesn't always mean that a gene behaves in the same way, as you may also remember from yesterday's Dr. Yeliz's presentation, but the use of animals in biomedical research has led to tremendous advances in the treatment of various diseases. On the other hand, the animal use for scientific purposes is reflected in the laws permitting but protecting their use in almost every country. Animal experiments deserve major ethical consideration, and such considerations should be in balance with the moral consideration in humans. Therefore, the concepts of replacement, reduction, and refinements as guiding principles for humane in vivo research was first proposed in 1959 as a strategy to address this responsibility. Since that time, the 3 R's concept, as you can also see here, has become widely accepted as a robust ethical framework for reducing animal use and suffering, helping to address societal concerns about animal research. So what are these 3 R's stand for? Replacement means that please replace the use of animals with non-animal methods where possible. If not possible, reduce the number of animals used to a minimum while still obtaining scientifically valid results. In addition, refine the practices to minimize the stress and improve the welfare of study animals used in the regulatory process. So in this light of these 3 R's, to create a model in science is about producing a simpler version of something more complex. Then you can see also here biological complexity. So it offers us to use less biological complex model system to understand more complex organism. So researchers can use a variety of model systems spanning from simple in vitro cell lines to large animals. All organisms are connected to each other through evolution, like members of a great big family. We share more or less characteristics of how we function according to the degree of relationship. The most basic characteristics are more widespread, while the subtleties are shared only with closer relatives. By using these evolutionary connections and evolutionary closeness, a variety of model systems can be used to infer rare disease mechanisms, which I am also interested in, or identify gene networks and therapeutic targets, and to test drugs, which I mentioned in my presentation in the last slides. Each model system has both advantages and also pitfalls, and model choice depends on a careful assessment of model's phase, construct, predictive, and target validity. In the concept of this presentation, as you can guess, we will focus on zebrafish and its biological advances. Although the common ancestor of humans and zebrafish swam into oceans more than 400 million years ago, they share many genes that makes the researchers a zebrafish enthusiastic on biomedical research. So, what is this zebrafish and where does it live in nature? So zebrafish were first described by a Scottish physician, Francis Hamilton, in 1822, and it's a family member of cyprinids, which is called as Danio rario in Latin. So the zebrafish is a tropical fish native to Southeast Asia, formerly described in the state of Bihar in northeastern India, but zebrafish have also being collected in the south and west of peninsular India, Pakistan, and also you can hear Bangladesh, possibly in Myanmar and Nepal. Then you can find these zebrafish in nature. Their typical habitat consists of shallow, slow-moving streams and particularly still pools that form alongside streams during the monsoons. The zebrafish is about 2.5 to 4 centimeters long, and in its larval stages, it is transparent. And as it matures to an adult, it develops stripes, as you can see here, along their body, and look in blue color. And as you can also imagine, this is why this fish called as zebrafish. And the fertilized eggs develop rapidly, extremely rapidly actually. Within 24 hours, all the major organs, as you can see here, all the major organs and tissues have formed within the transparent embryos, and within 3 days, the embryos have hatched into free-swimming larvae. Locomotor behavior begins with spontaneous muscle contractions from 18 hours post-fertilization, let's say HPF, and by 14 HPF, the embryos are responsive to touch that induces a rapid flipping movement. And after hatching, the larvae associate with hard surfaces by means of small secretory cells in the epidermis of the head. When they reach 4 DPF, or 4 to 5 DPF, they move to the surface of the water column where they inflate their swim bladders with air. Initially, for the beginning of their life, zebrafish larvae rely on yolk sac, this yellow part, rely on their yolk sac, nutrients for nourishment. But by 7 days, or by let's say 5 to 7 days, their yolk sac completely absorbed, and which means that you start to need to feed them, or they need to find prey for feed exogenously, to feed them exogenously. And larvae quickly develop distinct patterns of movement, including slow and burst swimming, routine escape turns, and complex behaviors that allow them to capture their prey. From around 14 to 29 DPF, larvae undergo a metamorphosis, and juvenile features such as scales are become acquired. And also aggressive encounters occur and shoal cohesion increases. At around 3 months, the juveniles become sexually mature adults, and spawning can occur. Courtship behavior is displayed from around 40 DPF when males begin to chase females. Females respond to this behavior by swimming alongside males for halting and presenting their flanks to them. And by 90 DPF, most females spawn eggs and most males attempt to mate. And the life cycle of zebrafish goes on like this way. Well, what are the advantages of zebrafish that make them move from nature to the laboratory as a very valuable system for biomedical research? First of all, the zebrafish are small and robust organism, which makes them easy to care and maintain in the laboratory conditions. Secondly, in contrast to many other fish, zebrafish are not seasonal breeders. Thus, embryos can be obtained for study throughout the year. Break of daylight triggers mating in zebrafish, while many other fish only lay eggs in the dark. The zebrafish has a generation time of 2 to 3 months. The zebrafish can produce more offspring in a shorter time, let's say 200 eggs or embryos per week. And in addition, fertilization and all subsequent development process occur within the water, not inside of the mother, which we call as external fertilization and development. And besides, zebrafish embryos doesn't need any parental care, or let's say minimal parental care, and all the developmental stages occurs externally. And besides, zebrafish embryos and early larvae are nearly transparent. Thus, using appropriate microscopic tools like microscopic methods like a confocal microscope, it is possible to image essentially every cells during the early stages of development. So, as I also mentioned, zebrafish embryos developed very rapidly, hatching a predatory larvae in 3 days, and thus the development of individual cells or structures deep inside the zebrafish embryo can be observed in real time, or by time-lapse videography as they occur. In addition to this, zebrafish offers an important advantages as a genetic model organism with its completely sequenced genome. The virtually transparent embryos and the ability to accelerate genetic studies by gene knockdown, knock-in, or overexpression studies have led to the widespread use of zebrafish in the detailed investigation of vertebrate gene function, the study of human genetic diseases, and increasingly drug discoveries. In the haploid state, the zebrafish genome has a size of about 1.4 gigabases distributed among 25 chromosomes, and detailed annotation provides evidence of more than 26,000 genes, protein coding genes, which is actually the largest gene set of any vertebrates so far I know sequenced. And comparison to the human genome, zebrafish genome, approximately 70% of zebrafish genome have at least one ortholog, and 84% of these genes known human disease-causing genes, which means that 80% of these 70% genes are disease-causing genes in humans. And let's say zebrafish embryos, let's say as a last advantage, zebrafish embryos are outstanding in their macroscopic size, and ease of injection also makes them exceptional system for manipulation and analysis of not only for cellular molecular biology, but also the genetic control of development. So we do not know the exact reason, but possibly in line with some of these advantages it has, in 1981, George Streisinger chose zebrafish as a model in which to explore the genetic basis of zebrafish embryos for vertebrate neural development. Although PubMed lists papers studying zebrafish embryos as early as 1950s and 60s in biomedical research, zebrafish research didn't hit its stride until nearly a decade after publication of pioneering work by Streisinger, and this is why he called as the father of zebrafish research. In the early 19th, two major laboratories decided to undertake a large-scale mutagenesis project in zebrafish to generate a panel of fish lines carrying recessive point mutations, affecting most of the genes involved in vertebrate development. And together with her team, Christian Nusslein-Volhard hit the first large-scale mutagenesis in zebrafish. In 1995, Thomas Hook took the advantage of small, transparent zebrafish embryos and larvae to observe the growth of tumors and to see whether they shrink in response to experimental treatments for cancer. And all these and similar highly important milestones not only catch the attention of researchers, but also the journals and their cover page. And since 1981, increasing number of researchers and laboratories have started to attract their attention on zebrafish. And nowadays, there are more or less 1,500 zebrafish labs all over the world. These labs are caring, maintaining, breeding, generating, and creating zebrafish to understand hundreds of biological mechanisms. Especially following with the improvement of genetic technologies from sequence to genome editing, the number of published articles started to increase each year. In 2020, or we can call it as the year of COVID-19, besides COVID-19, more than 4,000 research papers recorded in PubMed. And we are just in the fourth month of 2021, and approximately 1,500 articles have already been published. Yes, too many researchers are enthusiastic about the zebrafish. And the other question, what are the researchers doing with the zebrafish? Or I can ask, what do you want them to work? Zebrafish researchers mainly focus on investigating the cellular, molecular, and genetic mechanisms to underlie not only developmental process, but also rare human genetics, as I am trying to do, or cardiovascular disorders, or neurological and psychiatric disorders, or regeneration, cell signaling, cancer, drug screening, and many more. The high degree of conservation between zebrafish and humans, coupled with the advancement in genome editing technologies, have greatly expanded the number of zebrafish models of human diseases. So since the high DNA cutting efficiency and the flexibility of CRISPR-Cas9, which is when compared with the other methods, and which is also brought the Chemistry Nobel Prize to the inventors in 2020, is more widely used nowadays to modify the genes and the genome. So, which also we have enthusiastic as much as we have in Zebrafish. So let's have a closer look to how we can make rapid and efficient mutagenesis using CRISPR-Cas9 genome editing in Zebrafish. This is a very brief explanation of the protocol that can be used to generate mutations in a gene of interest. So first, you prepare a guide RNA oligos and Cas9 to inject your embryos and test genotyping primers and assays. So injections need to be done at 1 cell stage or maximum 2 cell stage, but at 2 cell stage you need to inject both of the cells, and this injection will generate double-stranded breaks during the first few runs of cell division. And the repair machinery is prone to errors, and those cells will carry a different type of mutation, giving a range of insertion and deletion mutations, which cause mosaicism in the individuals. The overall mutagenic efficiency is typically high, and we can say around 25 to 80% with fragment analysis. And if you use transgenic lines carrying fluorescent proteins, for example, you can check these fluorescent under the microscope. For example, you can check the efficiency of injection under the microscope in addition to mutagenesis and assessments such as melting or fragment analysis or sequencing analysis. These methods will determine a quantified mutagenesis rate, which can be correlated to a phenotype. But so here you can also see how we can follow up the process. So note that when you inject the F0 1 cell stage embryo, and the fish will grow up into more or less nearly 3 months. So you need to find the fish who carry these desired mutation in their somatic, in their gonads, not the somites. So you need to cross them with a wild type to see which of the zebrafish can carry these mutations in their gonads, so that they can create embryos and their new generations carry the mutation in their genotype. So if you want to, so you need to avoid mosaic mutants, which are called as crispons, can also be grown up, will also be grown up. So to see the phenotype, you can also see the phenotype, but it's not so reliable. So you need to get F2 generation for the desired phenotype. To generate a stable mutant line, CRISPR-Cas9 injected F0 individuals carry mosaic mutations defined by sequencing analysis of a fin clips can be crossed for first outcross to F1 to get F1, to outcross to wild type to get F1, and then you need to cross them both to wild type, or if you want to get homozygous alleles to get the homozygous mutant phenotype, you need to cross F1, you need to incross to F1 lines. And F2 homozygotes for recessive alleles for functional analysis, you need to F-line homozygous, which means that if you want to perform functional analysis regarding to the phenotype that you desired or inspected, you need homozygous F2 generations. At the end of more or less 9 months, you will have zebrafish within the phenotype, with the phenotype that you desired. And what you can do next, as a next step, is to discover a drug candidate to rescue the phenotype, for example. And with the advancement of next generation sequencing by means of whole exome or whole genome, and many genes become a candidate gene for a specific phenotype. To prove these variants, many genes can be a candidate for your patient. So you need to be sure which is a real causative gene for a specific phenotype. To prove these variants, whether you need to find more than one patient or you need to do functional analysis to prove the relationship between the variant and the disease. You can also find variants from cohort or large GWAS studies. And when this is the case, Zebrafish can provide a primary testing platform, both genetically and pharmacologically. Zebrafish provide two experimental arms, as you can also see here. And these two boxes allow simultaneous drug target validation. This figure is from an osteoporosis zebrafish model. So they try to find osteoanabolic compounds to rescue the osteogenic phenotype. So this is why you can see the bones and tails of zebrafish. And when you find an efficient drug candidate, you definitely need to test these drugs in mammalian model system as a preclinical trial. In this stage, Zebrafish provide you test more drug candidates in a very limited time and in hundreds of individuals. When you remember that these one pair of zebrafish adults can give you hundreds of eggs per week. So definitely shortens the time when compared with the mammalian drug screening part. So zebrafish can also assist you in all drug discoveries actually and development phases. Fundamental and preclinical research using zebrafish generate new targets and help to validate predicted targets in vivo. Cornerstones of the traditional research and developmental pipeline, including lead optimization, toxicology, testing, and long-term efficiency, as well as safety studies, are being developed in preclinical zebrafish assays. Incorporating whole organism testing into traditional research and developmental pipelines early in the process can help critical decision-making and avoid late-stage failure of new chemical entities. So in the genetic era, or we can clearly say that in the era of high-throughput omic technologies, the unprecedented amount of data that can be generated presents as a significant challenge, and especially for oncologists trying to provide their patients with the most suitable treatment for their cancer, for example. At this point, zebrafish get on the stage again and combine the characteristics of cell lines and animal models. Therefore, genetically engineered zebrafish models, and maybe we can say xenografts, are currently used for drug screening and preclinical drug development. And what is expected in the near future is to insert tumor grafts in zebrafish embryos could be used for patient selection in clinical trials. A genetically engineered zebrafish could be used to fill the gap whenever a mouse model does not exist. And besides this, xenotransplantation technique can also be used. You can inject patient-derived tissue or tumor cells into hundreds of zebrafish embryos, and the tumor-bearing embryos can be treated with a drug or drugs of interest, the cocktail of drugs that you interest, and for example, the chemotherapy that your patient is receiving. After 3 to 5 days, some embryos will have cells that have intravasated and migrated to the tail in a process that has similarities to metastasis. And some cells will remain viable at the injection site in the yolk sac. So the subset of cells that are resistant to drug treatment can then potentially be isolated, and subsequent mutation and gene expression profiling of these cells can identify potential drug targets. Or let's say none of the chemotherapeutic drugs helped your patient, and you need to discover a drug work efficiently in your patient, a personalized drug, where the future medicine is moving actually. Again, you can start injecting tumor cells to the zebrafish embryos. and tumor-bearing zebrafish embryos are arrayed in 96 well plates and threatened with threatened with multiple drugs. Then tumor-bearing zebrafish embryos then imaged and then treated with a selected potential chemotherapy based on molecular profiling, tumor board input, and the judgment of the treating, of course, the oncologists. So the tumor cells behaviors, including the proliferation, angiogenesis, migration, and metastasis, are evaluated over a 3 to 5 day period, which is really very fast when compared with the methods that we have now. So based on the effects of the tested drugs or drug combinations on the selected behaviors, a chemosensitivity profile for the tumor of the patient can be developed, and oncologists can select which is suitable for their patient. But of course, the drug sensitivity assays must first be validated in mouse models to ensure similar sensitivity to the drugs. But when these methods become much more reliable, in the future, it is expected that in less than 7 days, the oncologists will be armed with potential actionable information to guide treatment decision. So, to make a long story short, the zebrafish comes close to being the ideal model organisms for vertebrates, not only for developmental process, but also for drug discoveries, because it appears to combine the best features of all other models. It is estimated that one day a fish could help to save someone's life in the genomic era. This is not just a dream, actually. And with all of my enthusiasm for zebrafish, I can clearly say that zebrafish will not only extend our molecular and biological understandings, but also will be used to identify the best treatment option in the near future. And thank you for your attention, and I'll be happy to answer your questions if you have any. And if you have any questions, you can always get in touch with me, Elanur Yılmaz, at ku.edu.tr.
Thank you so much, Dr. Yılmaz. We only have one question, please.
Yes.
Can we use microinjection of SCR7 or RS1 to improve CRISPR's HDR knock-in technology in zebrafish?
Sorry, can you repeat the question? I couldn't catch.
Yeah,
I can see the question right now. Sorry. Can we use microinjection as CRISPR HDR in zebrafish? Actually, I'm not very familiar with SCR7, but you can check by using Zephin, a Zebrafish International Network, that they can list all the labs around the world, and you can check whether if there is any SCR7 injected phenotype. But definitely nowadays, what I know is that more efficient CRISPR technologies moving on, both to activation or deactivation or reactivation of the genes, which is very new actually. And then you can increase the efficiency of CRISPR, and then you can get the phenotype in F0 line, which means that you can get the phenotype in 3 months. So I'm not 100% sure for right now, but I'm quite sure that we can get a very highly efficient CRISPR in the very near future.
Thank you, Doctor. Doctor, there's another question about,
yes,
are there ethical issues in using zebrafish as a model organism in clinical trials or genetic engineering researches?
Yes, all the animal experiments need to be done in ethical issues. So first you need to take your ethical certificates to apply a course, and to take your ethical experiments usage certificates. So you cannot perform any experiments on animals as the way you are. First, you need to get your certificates, and then you need to apply ethical committees that you want to perform, why you need to do that experiments on that animals. But first you need to be choose that you will use the least biological complexity animals for your research. So it is very, very important, and you definitely need to get the certificate and also the application of ethical committee reports.
Thank you so much, Dr. Yılmaz. We're done for today.
You're welcome. Yes, thank you.
Dear participants, we'll be taking an hour break from our symposium and will be resumed at 1 p.m. according to Cyprus and Turkish time.
Catching a bus, never nothing when it's real.